Good morning, everyone. I'm Jim McCracken. It's a pleasure to introduce today's speaker in our Child Grand Round series. And it's a special delight to introduce my longtime colleague, Professor Robert Asarnow, for today's talk. Dr. Asarnow obtained his PhD at Wayne State University, spent time at Waterloo, Ontario, where he began what emerged as seminal work in the area of psychosis endophenotypes and associated familial characteristics. Bob is currently the Della Martin Distinguished Professor of Psychiatry and Behavioral Science and Distinguished Professor of Psychology. I believe Bob, in fact, was the first professor in the child division to achieve the exceptional rank of distinguished professor. He's been involved in three major research foci over the years. Childhood schizophrenia, which he's widely recognized as an international expert. Since then, he's moved on to add additional areas of interest, including traumatic brain injury in children and adults, applying translational research paradigms, as well as clinical observation description. And likewise, his third foci has been looking at the impact of surgical brain resection for intractable pediatric epilepsy, a broad portfolio of interest indeed. Today he's going to talk about his research in the childhood onset schizophrenia, a very rich and fascinating story. We're going to be using the question and answer function, so please submit any questions that you have to the Q&A box, and we'll have time for this discussion at the end. So, Bob, welcome and take it away.
Thanks so much, Jim. So, to begin with, I have no conflicts of interest regarding this talk to disclose. And to reiterate what Jim said, submit questions that you have as I go along via Q&A, and we'll get to them at the end of the talk. So what I'm going to share with you today is a program of research that I've been involved in for more years than I'd like to remember. And it focuses on neurocognition in childhood onset schizophrenia. The research has been supported over the years by grants from the Ontario Mental Health Foundation when I was at the University of Waterloo and McMaster University Medical School in Canada, 5 NIMH grants, support from the Della Martin Foundation, the MacArthur Foundation, and Janssen's Pharmaceuticals. I really am delighted to have this opportunity to share with colleagues who I haven't seen for the last 2 years what I think is a really interesting counterpoint to how research has been conducted in major mental disorders. One of the joys of being at UCLA isn't dealing with all of the administrative hassles, but having wonderful colleagues. And the work I'm going to present is an example of the multidisciplinary, across department, and actually across institution, different universities of research that has made being at UCLA such a special place. So the UCLA family study, which I'll describe to you, has involved many colleagues over the years. Keith Nectarline has played a particularly important role in this, and includes genetic collaborators from the University of Virginia. The work in cortical striatal circuits involves people both in the Department of Psychiatry and the Department of Psychology, and some wonderfully talented graduate students and postdocs, Jennifer Forsythe, Karen Ford, Dana Waxall, and Jessica Cohn. So this program of work that I'm going to describe really emerges from recognition of the challenges in identifying the pathobiology of schizophrenia. Of course, we want to understand the pathobiology in order to develop more targeted and more effective treatments for this devastating condition. As you all know, schizophrenia is diagnosed on behaviors. There are no laboratory tests we can use. There are no animal models, and much of what we know is based on the mechanism of effect of antipsychotic drugs. Fundamental enduring issue is that patients with schizophrenia are phenotypically and biologically heterogeneous. Schizophrenia symptoms are probably final common pathways for multiple brain abnormalities. So in the course of my career working in schizophrenia, I've seen excitement about multiple silver bullets. There was a schizophrenogenic mother. And then as we became aware that schizophrenia is a biological condition, multiple biological factors have been explored, and including prions. So, so far, none of these silver bullet approaches have really worked. And in response to that, numerous investigators have developed alternative approaches. The one approach that I've pursued has really, I think, two cardinal characteristics. The first is to study childhood-onset schizophrenia with the belief it may be a more familial, homogeneous, severe variant of the disorder in which the biological substrate may be more discernible. The second aspect of the approach was focusing on elucidating the pathobiology of endophenotypes rather than focusing on schizophrenic symptoms. So just to remind people, childhood onset schizophrenia, as you all know, is a disorder of late adolescence, early adulthood, and childhood onset schizophrenia is a very rare variant.
Dr. Asarnow, we can't see your slides.
Oh, okay. We, some years ago, confirmed earlier observations in a rigorous study, which I'll tell you more about shortly, the UCLA family study, showing that there was an increased aggregation of schizophrenia and schizophrenia spectrum disorders in family members, first in the parents of schizophrenia, childhood onset schizophrenia probans. For the narrow spectrum, there is a rate of almost 14% in parents of children with schizophrenia, compared to a rate of 5.4% of parents of children with ADHD, and less than 1%, which is about the population base rate in a group of community controls. In addition, we discovered, and Dave Fogelson has written a number of papers on that, that avoidant personality disorder tended to aggregate in families where the patient had childhood onset schizophrenia. So there was some support for the belief that childhood onset schizophrenia is a more familial and more severe variant. The second aspect of the approach is to look at endophenotypes. So endophenotypes, as defined by Dave Braff et al., are quantitative, heritable. Are my slides not showing? I'm so sorry. These are my collaborators. And this is the data from the family study showing the familial aggregation of schizophrenia and schizophrenia spectrum disorders, showing there's an increased rate of those disorders in families of parents where the proband is a child with schizophrenia. And then endophenotypes are quantitative, heritable, trait-based deficits, usually assessed by laboratory-based methods. They're seen as closer to genetic variation than the clinical symptoms of schizophrenia. Because they're more proximal to gene expression, they can facilitate examination of gene-environment interaction. Endophenotypes actually have been studied in a wide variety of complex medical conditions. Colon cancer, in colon cancer, multiple polyp formation rather than cancer is the genetically inherited trait. Studies of type 2 diabetes have focused on endophenotypes. In our recently funded grant to identify biomarkers of persistent concussion in adolescents, we're attempting to identify endophenotypes, one of which is involves autonomic dysregulation. These are the criteria for endophenotypes that Gottesman and Gould articulated. An endophenotype is associated with illness in the population, is heritable, is primarily state dependent. It manifests in an individual when the illness is not active. It, in their initial formulation, which is really based on linkage analysis, they wanted to see co-segregation of illness and the endophenotype, and that isn't really necessary. In fact, you'll see in our work that there's some independence, which is really interesting. The endophenotype found in affected family members is found in non-affected family members at a higher rate than in the general population. So what I'd like to share with you now are a series of studies that my colleagues and I, initially in Canada and later in UCLA, have conducted to identify neurocognitive phenotypes. Neurocognitive impairments are present in many patients with schizophrenia. Importantly, schizophrenia is a disorder of brain connectivity, and studying neurocognitive impairments provides insight into abnormal brain circuitry. It can be studied in animal models, and brain circuits supporting a cognitive process can be mapped with brain imaging in humans, and I'll give you an example of that towards the end. It's important to note that other endophenotypes have been studied in schizophrenia, including smooth pursuit eye movement, pre-pulse inhibition of startle response, and P50 suppression. So I started off this work focusing on a task called the span of apprehension. It's a task in which arrays are 1, 3, or 5, or 10 letters are presented tachistoscopically for 50 milliseconds, because that's faster than the saccadic reaction time. And so what you're measuring is the amount of information that can be scanned in iconic memory. So this task requires rapid scanning of iconic memory before it decays. One of our original studies was to look at this measure in groups of both acute schizophrenics and controls. And you can see here that patients with schizophrenia, both acutely disturbed or in clinical remission, showed deficits on this task relative to matched controls. They showed a reduced rate of early visual information processing. We replicated these findings and showed some diagnostic specificity by comparing patients with remitted schizophrenic patients to patients with bipolar disorder. And actually, one of the more important findings was that while there were group differences between patients with schizophrenia and patients with bipolar disorder, those differences were driven by a subgroup of patients. And that's really a critical, important point. In complex diseases, there are almost invariably multiple endophenotypes. But the advantage of this approach is that you can focus in a precision medicine kind of way on an endophenotype to identify its pathobiology. And we discovered that these deficits in the span of apprehension test can be found in children at risk for schizophrenia. So these are normal adults. These are primarily controlled children. And what we did was, with the data we collected from children whose biological mothers were schizophrenic, we subjected the data to a type of cluster analysis where we classified people on the basis of their attentional performance. And what we discovered, there was a subgroup of kids, cluster 4 children, whose performance was identical to that of acute and remitted schizophrenics. So this was a group of kids who had never been schizophrenic, but whose mothers were, who showed the same deficits as kids, as adults with schizophrenia. We then went on, I collaborated with a group at the Karolinska Institute who were conducting a study of normal aging. And we looked at monozygotic twins reared apart, reared together, and dizygotic twins reared apart and reared together, a kind of classical design for determining the heritability of the trait. Now, keep in mind, this is in people who aren't schizophrenic. And what we found was that a substantial part of the variability in performance on this task was under genetic control. Part of it shared with IQ, part of it unique. About 20 years ago, Keith Nectarline and I started the UCLA family study. And there were 2 arms, child and adult onset schizophrenia. Keith was the PI of the work on adult onset schizophrenia. I was the PI of the work on childhood onset schizophrenia. We studied childhood onset schizophrenia, attention deficit hyperactivity disorder, and community controls. And in this phase of the work, we used 3 attentional tests. A trail-making test, which is a widely used neuropsychological test, a continuous performance test in which the stimuli were perceptually degraded. This was a task that Keith developed for his doctoral dissertation, and the span of apprehension test. So in these analyses, what we did was we combined the data from community controls and ADHD mothers, and the same for fathers. And we used a logistic regression approach where we combined the scores from these three tasks. And what we discovered was that the scores on these three tasks identified about 22% of the mothers of patients with schizophrenia and none of the controls. And we obtained similar findings with in the fathers of childhood onset schizophrenics. So non-psychotic parents of childhood onset schizophrenia probands perform more poorly on a combination of tasks than parents of ADHD and community control probands, and a substantial proportion of the mothers and fathers performed outside of the range of parents of ADHD probands. And what was particularly interesting was that the presence of these deficits, neurocognitive deficits, in mothers was independent of the presence of neurocognitive impairments. So that these neurocognitive impairments weren't simply reflecting subtle, for example, schizophrenia spectrum disorders. Many other cognitive tasks have been studied in non-affected relatives of patients with schizophrenia. This is from a meta-analysis by Snitz et al. And you can see that actually the effect sizes become quite substantial. So it turns out that it isn't actually extraordinarily difficult to demonstrate that non-affected family members of patients with schizophrenia have subtle neurocognitive impairments. That creates a bit of a conundrum. These tasks tap a wide variety of brain circuits, and it becomes quite a challenge to find some common element in the many tasks that detect neurocognitive impairments in first-degree relatives of patients with schizophrenia. In addition to demonstrating the presence of impairments, endophenotypes should, as according to Gottesman et al. And Braff et al., be heritable. And so here I show you data from the UCLA family study, where we just looked at a wider range of cognitive tasks. This is in the second phase of our work, where we had a much more extensive battery, and we reduced the battery to a manageable number of factors. And what we discovered is that some of the performance was highly heritable in families of AOS parents, adult onset parents, and some was heritable in families of childhood onset schizophrenic families. And there was a difference to some extent in terms of what factors were heritable, showed familial transmission within families of adult onset and childhood onset schizophrenic factors. And what I found particularly intriguing was that facial memory showed familial transmission in childhood onset schizophrenia, not in adult onset schizophrenia families, because there's as probably many of you know, a substantial genetic overlap between childhood onset schizophrenia and autism. So these neurocognitive impairments are present in patients. They're independent of state. They are heritable. They show substantial genetic transmission in the normal population. They show familial transmission in families where there's either adult or child program with schizophrenia. And this comes from the work of Greenwood and Dave Braff from the Consortium on the genetics of schizophrenia. These are adult onset schizophrenia. And what I just want to show here is that you can see that down here are some neurocognitive types of endophenotypes, and these are electrophysiological phenotypes. And that the dotted line here represents a log score that exceeds chance levels. There are important role for genetic transmission, some more so than others, and different endophenotypes seem to be associated with different genetic regions. Here's from our study, the UCLA family study. What are in blue here are findings from the COGS sample that we replicated in our much smaller sample. And this was really the first iteration of our attempt to find associations between cognitive factors and genes. We started off with 1,500 SNPs in this analysis, and as you'll see later, we have regenotyped the sample and are looking at a much wider range of SNPs. In these initial analyses, Judy Rappaport and I discovered, in collaboration with Stefan Sanders and Matt State, a number of de novo mutations. This is the de novo mutation we found in the childhood onset sample at UCLA. Polynucleotide kinase phinase phosphatase gene. It's associated with microcephaly and plays a role in DNA repair. And it's been shown to be a neurodegenerative phenotype. So we found one of the patients in her childhood onset schizophrenia had this genotype. So we're going to be doing exome sequencing in a larger sample to see if we can identify additional copy number variations. And this is a summary of the genetics of childhood onset schizophrenia that Jen Forsyth and I conducted a number of years ago. And what we found from reviewing the literature is that childhood onset schizophrenia shows considerable overlap with adult onset schizophrenia. And the causal genes for COS seem to both involve common and rare genetic variants. It looked like there is a greater familial aggregation of schizophrenia spectrum disorders and a higher rate of copy number variations than in adult onset schizophrenia. There's considerable genetic overlap with autism. So, as I mentioned in the beginning of my talk, one of the potential advantages of focusing on neurocognitive phenotypes is that they become a platform where you can drill down to identify the underlying processes. And so in following that up, what my colleagues and I thought might be a productive approach was to try to identify simpler cognitive functions, simpler than the neurocognitive tasks that we'd used before. The belief was that simpler cognitive processes had more circumscribed circuits. They were less widely distributed, and they could provide leverage in linking functional genomics to circuits associated with liability to schizophrenia. There, however, is a cost involved in doing this. As probably many of you are aware, the current functional GWAS studies of schizophrenia entail hundreds of thousands of patients who are for the most part where the phenotype is the presence of schizophrenia. And that permits you to really conduct large-scale studies all over the world. In contrast, studying these neurocognitive impairments, while there are virtues in deep phenotyping, greatly reduces, by orders of magnitude, the sample size. So we decided to study procedural or habit learning, which is a type of learning that's been very well studied in animal models, where you have a gradual learning of stimulus-response associations over extended trials. And this type of learning is supported by circumscribed circuits involving the frontal lobe and the basal ganglia. So there's been long-standing belief that pathophysiology of schizophrenia is linked to dysfunctional cortical striatal symptoms. There's structural abnormalities in the basal ganglia in patients with schizophrenia. And there is beautiful models of cortical striatal interactions, as well as animal models of habit learning. The circuit that we're going to focus on involves the putamen and the dorsolateral prefrontal cortex and the ventral striatum. And our first stab at this was to conduct a case control study of patients with schizophrenia and to compare their performance on two tasks, a habit learning test, which I'll show you in a second, and a serial reaction time, which is a motor skill learning test. And this is the habit learning test. This is Mr. Potato Head. And the way the task works is that Mr. Potato can have various types of little features. And these features are vary in terms of the probability they're associated with the outcome. So for example, vanilla and chocolate ice cream. So if you're the participant, you see Mr. Potato Head in one of these screens, and you have to guess whether he's going to get chocolate or vanilla ice cream. And none of the cues is perfectly linked to the outcome. And there are a variety of the cues. So what people are learning are the probability of a set of cues being associated with an outcome. And this is the serial reaction time test, which involves a kind of motor learning. And what we discovered is that patients with schizophrenia, these are adult onset schizophrenics, showed remarkably poor performance on this task compared to controls, both when the task was presented by itself or when there was another task presented as well. And so we showed that habit learning is impaired in patients with schizophrenia, but we were worried that since the patients with schizophrenia we studied were patients who were receiving antipsychotic medications, and there was extensive literature that antipsychotic drugs frequently result in enlargement of basal ganglia, we were concerned that maybe what we were looking at was the effect of antipsychotic drugs, on patients with schizophrenia. So our solution was to study the unaffected relatives of patients with schizophrenia. So we developed a slightly different kind of habit learning test. The same principles applied. There were a set of stimuli that were presented that were probabilistically associated with an outcome, sun or shine. And this is called the weather prediction task. And here you can see very early in practice that the siblings of patients with schizophrenia perform more poorly than controls. Controls, as it turns out, very quickly start to associate certain highly probabilistic cues with the outcome, and the patients with schizophrenia don't. We then did a series of brain imaging studies, of which I'll show you only one, using fMRI to measure brain activity while siblings of patients with childhood-onset schizophrenia performed the weather prediction task. And what we discovered was that, contrary to what we expected, the siblings of childhood-onset schizophrenia patients showed increased activation of multiple brain regions compared to controls early in practice. However, after extended practice, siblings of childhood-onset schizophrenia proband showed reduced activation compared to controls. So you can see that here, the yellow areas are where we found reduced significant differences between performance in a baseline condition and the practice condition in patients with schizophrenia, where the difference between baseline and the practice condition was greater. So patients with schizophrenia showed greater activation in these regions. And as you can see, some of these clearly involve basal ganglia. And then there was and here we find the opposite results. So where do we go from here? So what we're currently working on are genotyping the UCLA family study using the global screening array chip approach from Illumina. And we're comparing the genetic architecture of childhood-onset schizophrenia and adult-onset schizophrenia using the global screening array data and measures of copy number variation burden. We're examining the association between neurocognitive impairment and genes previously linked to early neurodevelopmental disorders and brain development. Jen Forsyth is playing a key role in that. And we're generating polygenic risk scores for IQ and schizophrenia to determine the extent to which the genetic architecture of different neurocognitive endophenotypes overlaps with IQ and schizophrenia. So this has been a very interesting journey. I think what this has gotten us to is a place where we can ask questions that are more likely to result in answers, shedding light on the pathobiology of schizophrenia, but there's a long way to go. And with that, I will turn this over to Jen.
Thank you so much, Bob. I think one of the things that your presentation demonstrates is your dogged persistence in the search of knowledge and scientific goals, and you're to be applauded for that. But yeah, so many fascinating findings along the way. At sort of a macro level, one of the things that comes through to me is the importance of studying childhood onset forms of psychopathology and comparing them directly as you did to adult forms, and how revealing that can be, and or how many new questions can emerge from such experimental comparisons. Several things that popped out to me that I found really intriguing. One was the familial aggregation of avoidant personality amongst the family members, and other personality disorders as well. I'm wondering if you have any reflections or comments about that type of aggregation of characteristics that you uncovered.
Well, a couple of thoughts, Jim. One of the things that was interesting was that actually avoidant personality disorder occurred more frequently than schizotypal personality disorder, and that the two of them, these two personality disorders, really didn't overlap very much, to our surprise. The second is that there's, as you know, in autism, there's extensive literature showing that there's a dimension of the ability to form interpersonal connections that seems to be heritable, and that part of that heritability seems to be genetic. And I guess the third observation is from sitting in on making the diagnoses of the family members. Avoidant personality disorder, as we use the term, really wasn't shyness. These were people who really avoided interpersonal situations. So I think that it's an interesting finding. It is, in my opinion, harder to drill down on that than in some of the electrophysiological and neurocognitive types of findings.
Thanks. I thought the presentation of the data from the Greenwood linkage studies was really interesting. One of my takeaways to see if you agree or not, is that good old neurocognitive tests were more powerfully linked than electrophysiologic measures. At least it appeared to me that several neurocognitive measures achieved a higher LOD score, some in the range of 4 and above. So I was going to give a cheer for neurocognitive testing, if you agree.
Well, I have mixed feelings. My doctoral dissertation was looking at visual evoked potentials in schizophrenics with and without a family history of schizophrenia. And so I have a long devotion to using electrophysiology. But the neurocognitive measures are cheaper and lend themselves to larger scale data collection. Albeit not the large scale data collection that are currently being conducted in GWAS studies of schizophrenia. So they really have some advantages. The habit learning task that I became interested in, I think is really, really interesting in terms of providing opportunity to do translational research. So we, as you know, since you helped us out, looked at the effect of D-cycloserine on habit learning, because we believed that it was a glutamatergically mediated kind of process, and we found that we could enhance habit learning in controls, but unfortunately not in patients with schizophrenia. So you can do those sort of studies with these neurocognitive tasks.
Your genetics results were likewise intriguing, and I wasn't sure if you're yet in a position to make any estimates about the frequency of what you believe might be pathogenic copy number variants within your samples, or if there's any other data out there for childhood onset schizophrenia.
There is. So what I showed was some preliminary data from a poster that one of Matt State's postdocs presented, and it was data from our sample and Judy Rappaport's childhood onset schizophrenia sample. But right now we're actually regenotyping our entire sample, and in the process of that, we'll have a much closer look at copy number variations. What seems to be the case in schizophrenia now is that there is a kind of balance between the liability caused by copy number variations versus common genes. And I think in the case of childhood onset schizophrenia, more of that balance tilts towards the rare variants, the copy number variants, than in the more typical adult onset schizophrenia. But we'll have a better sense of that when we're done regenotyping.
I just want to give you a shout out that there may be some people in the audience who don't appreciate that there are only a couple of centers around the world that have recruited and studied samples of the childhood onset schizophrenia. So again, I want to applaud you for that accomplishment, and we can see the richness of the research that comes from that. The adult-child differences in neurocognitive familial transmission also was really, really interesting, and just seems to me to go further in supporting the premise that childhood onset schizophrenia has some key differences. I'm wondering if there's any way to kind of hypothesize or speculate about brain regional differences as far as cognitive dysfunction. Does it look like in your efforts to kind of synthesize what's more associated with child versus adult, that one could make any proposals about adult being possibly more frontally involved or prefrontally involved as opposed to the childhood form, or any kind of interpretation of that sort?
You know, it's a great question, Jim, but I really don't think we have an answer. Neither Judy's group nor our group really compared brain imaging directly between patients with child and adult onset schizophrenia. I think, you know, just sort of segue, one of the unexploited questions are areas of inquiry that could be pursued with childhood onset schizophrenia is, why is there childhood onset? Why do some people develop schizophrenia very early in life? And Jen Forsyth obtained the K Award, which she's now pursuing as a faculty member at the University of Washington to begin to address that question. That in some ways, I think, is really for me, the most interesting question. It may simply be that there's a greater load of deleterious genetic variants, or it may be that there are some kind of protective factors that in childhood onset schizophrenia you don't find.
Thanks. If the audience has any other questions, please feel free to use the Q&A function. The other question that your work sparks to me is, what are the implications of your work with the childhood onset variant on helping us achieve a more sensitive or specific notion of what the prodrome of later onset schizophrenia may look like, to facilitate, you know, earlier and more accurate detection, or, you know, within that 20% or so of prodromal individuals who do convert. Does your work have implications for those kind of prognostic implications?
Well, actually, when I first came to UCLA, my work was supported by the MacArthur Foundation. And I was part of the MacArthur Foundation group that included Mike Rutter. And Rutter had published a seminal paper claiming that autism and childhood onset schizophrenia were distinct conditions. And there's a lot of context to that, which Jim knows, and maybe some of the younger folks do. Childhood onset schizophrenia had really been, prior to DSM-3, a grab bag diagnosis that included all sorts of weird neurodevelopmental abnormalities and autism. And so Mike really made a lot of progress by separating these. And he was really appalled when we published the paper showing that prior to the onset of schizophrenia, about 60% of these kids who subsequently developed schizophrenia had very profound delays in language acquisition. And he said, well, Asarnow, what are you trying to do? Undo all the progress I made. Well, you know, it's really interesting. Mike really made a great contribution, but what is it that we discovered in subsequent years? That actually there is a substantial biological and genetic overlap between childhood onset schizophrenia and autism. And maybe part of that overlap has to do both with the early history of language delays in childhood onset schizophrenia, which by the time the kids are adolescent in childhood onset schizophrenia really are no longer detectable. But the persistent problems they show in informing social interactions. And maybe that's where the overlap is. So, yeah.
Well, thanks so much for your presentation today, and congratulate you on all of this work that's so interesting and much appreciated.
Well, thank you,
Signing
Jim,
off.
and I'm looking forward to seeing you and the rest of my colleagues in person in the near future. Take care.
Absolutely. Bye-bye all.